Good morning, students. I am Dr. Nirmala Pragain, Assistant Professor Botany, MBGPG College, Haldwani. In today's class, I will be discussing about reproduction in bacteria, which is the Unit 1 of BSC I Sem Botany syllabus. So, bacteria were discovered by Antony von Leeuwenhoek. They are microscopic unicellular prokaryotes found everywhere, such as in air, water, soil, etc., and play an important role in our life. They help in decomposing the dead organic matter. They help in increasing the fertility of soil, as well as they are useful in various industries. They are, however, responsible for causing a number of diseases in man, animal, and plant. So, in this class, we are discussing about the reproduction in bacteria. So, first of all, what is reproduction? It is a biological process by which organisms produce their offsprings. In case of bacteria, there are two types of reproduction. First one is the asexual reproduction, and second one is the sexual reproduction. Asexual reproduction is a type of reproduction that does not involve the fusion of gametes. In asexual reproduction, a new offspring is produced by a single parent. In sexual reproduction, there occurs a fusion of gametes, but in case of bacteria, sexual reproduction is that method of reproduction where gene transfer occurs in case of bacteria, and it takes place by three methods: conjugation, transformation, and transduction. So, first of all, the asexual reproduction. It takes place by two methods: vegetative reproduction and spore formation. In vegetative reproduction, fission, budding, gonidia formation, and cyst formation are important. First of all, fission. Under favorable circumstances, when there is sufficient amount of water, nutrients, and favorable temperature, the bacterial cell divides to form two or more daughter cells. If the bacteria is cocky, then division occurs in any plane. If it is a bacillus or spirillum, then occurs in a transverse plane. Binary fission is a very common method of vegetative reproduction where the parent cell divides into two identical daughter cells. So the first of all, this is the binary fission where parent cell elongation takes place, division of nuclear material and division of cytoplasm of cell. And division of nuclear material is followed by a formation of septum. The DNA pulls towards either end of the cell. The bacterium pinches off at the center and forms a septum, which is a separating wall between two compartments of the cell. As the septum formation is completed, separation of two daughter cells occurs. So you can see here from the parent cell, two daughter cell formation takes place, and the daughter cell gets a suitable substratum. They starts to germinate and forms new bacteria. Budding is another common method of vegetative reproduction in bacteria where cell wall at or near one pole softens and thins, and protoplast is covered by a thin wall bulge. Parent genetic material divides and enters in the bulge, and this bud grows and separates by a constriction at the base. And after separating from the parent, this bud grows and forms a new bacterial cell. In segmentation, bacterial cell protoplasm undergoes subsequent segmentation, forming very tiny structure, very small structures called gonidia. And when the cell wall ruptures, these gonidia get liberated and germinate on getting a suitable substratum. Next one is the cyst formation, where the bacterial cell protoplasm shrinks and secretes thick wall around it to form cysts. These cysts are similar to the endo. Next method of asexual reproduction is the sporulation, where bacterial cells produce non-motile spores such as conidiospores, oidiospores, sporangiospores, and endospores. Conidiospores are produced at the end of specialized aerial branches called conidiophore, and they are dispersed by wind or some other means, and on getting suitable substratum, these conidiospores or conidia germinate and produce new bacterial cells. Second one is the oidiospores. In many actinomyces, what happens? Separation of body occurs throughout the length, resulting in the formation of oidiospores. You can see here these oidiospores, and when these oidiospores get suitable substratum, they start and germinate to form new bacterial cell. Next one is the sporangiosperms. They are again produced in bacterial like actinomycetes, where sporangia are formed, and inside this sporangia, the protoplast divides to form tiny sporangiosperms, and these sporangiosperms on germination produce new spore bacterial cell. Next one is the endospores, where the endospores are produced under unfavorable circumstances in some bacteria, and they are highly resistant to the harsh climatic condition and are dormant in nature. They are very resistant to extremes of heat and also resistant to the toxic substances, and on return of favorable circumstances, as I already told you, these endospores are produced under unfavorable circumstances, and when they get favorable circumstances, they germinate to form new bacterial body. This is all about the asexual reproduction in bacteria, which takes place by means of vegetative reproduction and spore formation. The next one is the sexual reproduction. As I already told you, in bacteria, this is a gene transfer, or we can say transfer of genetic material. No offsprings are produced during gene transfer. In bacteria, there occur only the horizontal transfer of genetic material. So, no vertical transfer takes place. It means from parent to offspring. So, there are three methods of gene transfer in bacteria. First one is the conjugation, transduction, and transformation. In case of conjugation, gene transfer occurs between two bacteria with the help of a conjugation tube. It means it requires physical contact. Second one is the transduction process, where the bacterial gene is transferred by a specific virus that infects the bacteria and called bacteriophage. The third one is a transformation, where a piece of DNA would be taken up by bacteria from its surrounding environment and incorporating this DNA in the recipient cell. So you can see here in all these three methods, there is no parent cell to daughter cell gene transfer. In vertical gene transfer, it occurs from parents to the offspring. So this type of gene transfer in bacteria is called horizontal gene transfer. Now we will discuss all three methods: conjugation, transformation, and transduction one by one. First of all, conjugation. It was discovered by Lederberg and Totem in 1946. Here, in conjugation, gene transfer occurs between two bacteria and it requires physical contact. One bacteria acts as a donor and other as a recipient. So we can see here, this is the donor cell and this is the recipient cell. In this donor cell has its own DNA and extra chromosomal DNA, and we can say plasmid. The bacteria which contains plasmid called F plasmid, and this contains a specific F factor or fertility factor, and this is designated as F plus cell. The recipient cell you can see here are the cells which lack this F plasmid or F factor. So cross occurs between F plus cell and F minus cell. F plasmid directs the synthesis of sex pili. This is the sex pili, and F plasmid directs the synthesis of sex pili, which is a cytoplasmic bridge between two bacteria and helps in the physical contact between the two bacteria. With the help of this cytoplasmic bridge, what happens? The genetic material is transferred from donor cell to the recipient cell. Once this contact is established, one strand of F plasmid is transferred to the recipient bacteria. When it is transferred into the recipient cell, in both donor cell and recipient cell, the second complementary strand of the plasmid starts synthesizing. At the end of conjugation, once the plasmid is transferred and replicated, this physical contact, here you can see this physical contact will be released, and both the bacteria containing copy of F plasmids. The result of conjugation is both the cells are F plus cells. Now, the recipient cell also contains the copy of F plasmid, thus it can also act as a donor and carry out the same cycle again. So, in conjugation, when a cross occurs between F plus cell and F minus cell, both the cells are F plus cell, and this recipient cell also contains the copy of F plasmid and also acts as a donor cell. So this is about the conjugation, which is the horizontal gene transfer in bacteria. Second one is the transformation. It is a type of gene transfer in bacteria where a piece of DNA would be taken up by bacteria from surrounding environment directly into the cell and incorporating this DNA in the recipient chromosome. The question is that how this piece of DNA comes in environment. So when any bacteria is lysed, it is going to release all its cell content including DNA into its surrounding environment. Generally, when a bacteria is died or lysed, its DNA fragments are present in the surrounding environment, and these DNA fragments, when come in contact with a competent cell, it can be taken up by these cell, competent cell. These DNA fragments are taken up by competent cell and also get integrated into its chromosome. Normal bacterial cell is not permeable to take DNA fragment directly. Only these competent cells are permeable to the DNA fragment and they allow the DNA fragment to enter into the cell. When such DNA fragments comes in contact with the competent cell, it will bind with the competent cell. And one strand of these DNA fragments will hydrolyze by the envelope exonuclease of the recipient cell. So what happens? Only one strand of this DNA enters inside the cell and this will find a homologous region with the recipient cell and it will get incorporated into the host chromosome. If it get incorporated in the host chromosome, this process is called successful transformation. Sometimes what happens if this piece of DNA did not find the homologous region of the host chromosome, it will be degraded. You can see here it will be degraded. It is degraded. So transformation will not occur and it is called as unsuccessful transformation. So what happens in case of transformation? The bacterial cell lies, naked DNA released into the environment taken up by the competent cell because its membrane is permeable to take DNA fragment. It is integrated with the recipient chromosome. So there is a gene transfer from one bacterium to the another bacterium. This is called as transformation. The third important method is the transduction, which was discovered by Gender and Lederberg. It is another method of genetic recombination where genetic material from one bacterial cell is transferred to the other bacterial cell by a specific virus called bacteriophage. It occurs as a result of error in viral genome packaging. The important thing is that transduction occurs as a result of error in viral genome packaging. Bacteriophage commonly cause lysis of bacterial cell which they infect. Such bacteriophage whose infection is followed by lysis is called virulent. However, many phages do not always lyse the host and are called temperate bacteriophage. Depending upon these two types of transduction may occur. First one is the generalized transduction and second one is the specialized transduction. Bacteriophage when infects the bacteria show two types of cycles, lytic cycle and lysogenic cycle. In lytic cycle, generalized transduction occurs and in lysogenic cycle, specialized transduction occurs. In lytic cycle, when bacteriophage infects the bacteria, this is the bacteriophage and this is the bacteria. When this bacteriophage infects the bacteria, it attaches itself to the bacterial cell wall, adsorbs it and injects its nucleic acid. Here you can see it is injecting its nucleic acid into the bacterial cytoplasm. The virulent phase DNA codes for enzyme that cause the fragmentation of the bacterial DNA and directs the host metabolism to synthesize more phage nucleic acid and phage protein codes. After some time, these phage DNA and phage protein assembles. You can see here the assembly of phage DNA and phage protein, and they are forming new phage. Occasionally, what happens during this assembly, a small piece of bacterial chromosome, this green color, becomes incorporated into some of mature phage particles. So here in green color you can see this is the bacterial chromosome. Here there occurs error in viral genome packaging. There occurs error in viral genome packaging. Upon subsequent lysis of the bacterial cell, new phages are released, and when such phages infects a new bacterium, so there may occur three situations. First one is successful gene transfer, another one is the unsuccessful gene transfer, and third one is the survival. So we will discuss all these situations later. Second is the lytic. After this lytic cycle, second one is the lysogenic cycle. Here what happens? In lysogenic cycle, lysis of host cell does not take place after injection of phage DNA. In lysogenic cycle, the phage will infect the bacteria, release its DNA, instead of taking over the host machinery, get integrated into the bacterial chromosome and replicates at the same rate as the bacterial DNA. The phage genome at this stage is known as prophage, and the bacterium that carries that prophage is called lysogenic bacterium because it is capable of being destroyed at a later time if this prophage becomes active and re-enters the lytic cycle. So this is the lysogenic cycle. Now, in lysogenic cycle and lytic cycle, we will again explain it here. So I already told you that there occurs in lytic cycle there occurs three kinds of situations: successful, unsuccessful, and survival. So what happens when when this bacterial DNA or the phage containing this bacterial DNA get integrated into the host chromosome? It is a successful gene transfer because it is recombinant bacterial cell. Now this is the phage, bacteriophage containing the bacterial chromosome, and when this bacterial chromosome enters into the new host cell, it get integrated with the host chromosome, and this is called as successful gene transfer. If it is not, and if this new entered bacterial DNA fragment degrades, the second situation is that if this newly entered bacterial DNA fragment degrades, it is called unsuccessful gene transfer. There is also a possibility of survival of this DNA fragment. It is neither integrated nor degraded. So this is the survival. It is called as partially diploid structure. So in case what happens in this process, It is called generalized transduction because randomly any part of bacterial DNA gets packed inside the viral coat. No specific portion of bacterial genome get packed. So you can see here randomly any part of bacterial DNA get packed inside the viral coat. No specific portion of bacterial genome get packed. So this is called as generalized transduction and it occurs in the lytic phase only. Second one is the specialized transduction, as I already mentioned about this lysogenic cycle. I told you that this prophage becomes active and re-enters the lytic cycle. So when this prophage re-enters the lytic cycle, as a rare event, what happens? The prophage is free from the bacterial chromosome. This prophage is free from the bacterial chromosome, and in doing so, it accidentally carries away an adjacent gene from the bacterial chromosome. So the excised prophage spontaneously regains its virulence and becomes active and infect the other bacterial cell. Now it becomes active and infects the other bacterial cell. Here it will infect the other bacterial cell. So when this phase the other bacterial cell, this phase infects the other bacterial cell, the bacterial genome also get transferred to the cell which they infect. Okay, so here you can see the infected bacterial genome with some part of phase DNA, with some part of donor bacterial DNA along with the host DNA. Three combinations are here. Some part of phase DNA, some part of donor bacterial DNA and some part of host DNA. So this is the specialized transduction. In specialized transduction, the bacteriophage transduces only those bacterial genes that are adjacent to the integrated prophage. So recipient bacteria has own genome, viral genome and donor cell genome. So this is called as specialized transduction. Only specific bacterial genes are excised which is close to integration site of phage DNA. That's why it is called as specialized transduction. So this is all about the transduction where the by the a virus called specific virus called bacteriophage is responsible for causing gene transfer. So gene transfer occurs with the help of a virus, a specific virus called bacteriophage that infects the bacteria and gene is transferred. So this is all about the sexual reproduction in bacteria. It occurs by means of conjugation, by means of transformation and by means of transduction. So this is all about the reproduction in bacteria, which is very important topic of your unit 1 BSc first sem. Thank you.